Can you see my screen?
Yes, sir.
What is there? Okay. So, good morning, ladies and gentlemen. My name is Adeotunde Michael, and this morning we have come to another educational opportunity. This time around at Om3ga Bio-Edutainment. My belief before today is that you must have had a prior exposure to bioinformatics, and that was exactly what you did at your HND1, that is at your year one. So, and that is why we want to be more practical, and that is why you are seeing on your screen bioinformatics in microbiology practical lab manual. So this is going to be a step-by-step guide with tools, not only tools, case studies, and a lot of images that is going to also go on in explaining one thing or another thing about whatever your text might be telling you. Don't forget, I want you to go with me to the second slide, and that is module 1. Just like I told you that this time around, I expected that every one of you should have exposed yourself to basic bioinformatics, and this is not the time to start telling you the meaning of bioinformatics, telling you the meaning of blast, telling you a lot of other basic things. But this time around, it's like I want you to see yourself as being in the lab that you want to start doing your activities. So the first module that you are going to do, don't forget that at the beginning of this lecture, I told you that every one of you are expected to come up with a project at the end of the day, and this project you are going to follow this instruction very very well, and that project will be graded for whatever might be your essential reasons for one thing or another. So module 1 is data retrieval. That is, how can you retrieve data, that is microbial genome sequences. So on your screen, the first thing is that you visit NCBI genome. This is time to listen and watch. So this is not the time to do the activities, but it is the time for you to listen, watch your screen, and follow the step. So you have to visit NCBI genome browser, and by now you know how to do that. The next thing is you are expected to search Escherichia coli complete genome. This time around, what you are going to use for your own project is not going to be Escherichia coli. You should be thinking of another microorganism. So everybody must choose your own microorganism. It's not going to be the same as you have on the manual. Don't forget that this is a manual. A manual means a guide. It's just like something that is going to show you the road. How can you go about it? So the next thing is you filter. By filtering, you mean, don't forget one thing that you should know by this time around, that once you retrieve any sequence at all, the first thing is to clean up your sequences. You clean it up, and you know where to clean it. Visit that website, just input your sequences there and clean it. So after that, you filter it at ref-seq plus the complete genome. That's what you are going to that's what you are going to put into your browser. Then step 4 is to download faster. You should know that this time around we will always work with faster format. Then the next thing is a case study of E. coli. Now, whatever results that comes out, although that is not here, it's not in the practical guide. After whatever you must have gotten, after you must have done that, I want you to go to ChatGPT, go and input it there and tell ChatGPT to explain that result for you as a scientist or as a microbiologist. What is the meaning of that result? Okay, so let's go to module 2. Module 2 is a sequence alignment using BLAST. Sequence alignment using BLAST. Don't forget the reason why there must be alignment. The reason why there must be alignment is that you have a sequence and you want to compare that your sequence with any other sequences that you have in the databases. So you could have like 15 sequences and you want to compare them with whatever. How closely are they to choose database? The database is going to be NR/NT, or you choose RefSeq. Okay, that is reference sequences. That's what that means. Then you click on run the blast and review e-values. Run blast and review e-values. So if you look at your screen, you look at this small guy. You look at what he's trying to do. Under this description, he has Escherichia coli. After that, he also has a Salmonella enterica. After that, Klebsiella pneumonia, and after that, Pseudomonas aeruginosa. So what you will do there, you can see maximum score. That is 7E minus 50. That is for Escherichia coli. And the e-value, you also look at e-value. You record them somewhere. And by the time you record them somewhere, when you want to do your interpretation, always do your interpretation with ChatGPT. So whatever results you must have gotten, go and paste it on ChatGPT and prompt ChatGPT to give you or interpret it for you. Okay, so and you document whatever might be the result. On module 2, that is about genome annotation using a tool that is called Prokka. So the first thing is you need to install Prokka on your system. I don't know if your smartphone can take that. I'm not sure because I've not run it on a smartphone before. So after that, you now run Prokka E. coli.fna slash outdir E. coli underscore annotation. You can see the prompt on the screen. So step 3, you just click on analyze.gbk.faa.ffn, then output. So the case study is annotating Mycobacterium tuberculosis for resistant genes. So that is Prokka, you are trying to annotate. We already have a complete genome of an organism. And when you have a complete genome of an organism, you can do the interpretation. You want to predict whether that organism has some resistance potentials. And that is what this Prokka is going to help you to do. And when you look at the right side of your screen, you will see that this has been run on a small laptop, and you can see. So whatever you are getting, always compare it with what you have on the visual, on the images by your side. And that's why I said this presentation is a practical, and you have a lot of real life application, that is a case study. Then we also took some time to generate some kind of images for you, so that even if you cannot if you are lost, you'll be able to be guided by the images. Okay, then module 4, that is AMR prediction using CAD and RGI. So some of you that are working on antimicrobial resistance as your project, you will understand what is CAD. CAD is comprehensive antibiotic resistance database, that is CAD. Okay, the first thing is you will go and visit CAD, that is you type CAD RGI on your browser. You can do that on Google. Just type it there, it's going to guide you. Click it, and whatever file, whatever sequences that you have, load it there. Upload your file and choose perfect and strict criteria. So after you must have chosen that, the next thing is to submit and review resistance genes. So this was done for Klebsiella pneumonia resistance profiling. So once you do that, that one is going to guide you, so as to know or predict whether the sequence you have is actually harboring a kind of resistance gene. Don't forget that in your theory class, I told you, and by now you ought to have known, that everything that takes place in an organism, including the way they infect, including their choice of host, including every activity about any organism, such activities we must have molecular basis of it. And that is the reason why the first thing to actually do is to predict in terms of sequences, predict whether at molecular level your organism has the potentiality to develop resistance in the nearest future. So by the time you have sequences and you are using bioinformatics tool for this, that one is going to guide your decision. It's going to guide you whether whether the microorganism you have will have antibiotic resistance. And not only that, it is also going to specify the set of antibiotics that such organism might develop resistance at. Okay, and that's the reason why they use CAD for that. There are other modern databases apart from CAD, apart from CAD. So you could, that is beyond the scope of this class. But if you actually want to know, you could also check out for all this. And don't forget what we are saying. What we are saying in essence is just that, number one, that is going to guide the treatment, is going to guide diagnosis. For example, now let's say somebody is sick, or if there is an outbreak, and in that outbreak you collect their samples. When you collected their samples, you do a lot of analysis in the laboratory, wet laboratory. What you do, you culture, you identify, you do whatever you want to do with it, and after that you do the DNA extraction. After the DNA extraction, you run the polymerase chain reaction that is going to amplify the DNA that you have. After amplification of that, you now do the sequencing, complete sequencing, to know the type of alphabet that that organism is having. And not only the type of alphabet, you also know the arrangement of those alphabets, because the way an alphabet is being ordered will definitely go a long way to tell us, to give us information about anything. For example, as I used to say, if somebody name is Kola, that is Kola, and if there is a misarrangement in the alphabet, you will see that it's not going to sound and it's not going to look like color any longer. So the same thing with all these microorganisms. The way they are being sequenced determines the what they are capable of doing. So by using CAD, so immediately after you get your sequences, you now run it. You run it on your laptop or your smartphone. You run it there, and the next thing is going to predict whether all those set of genetic codes that will be standing for resistance, they will tell you. It could be grouped into three. It could be perfect resistance, it could be strict resistance, or it could be loose resistance. In fact, you can even do that across the continents. You can do it across the continent. Let's say what you have is you already have the sequences. So when you have the sequences and you run it on CAD, that one will tell you. If you want to know whether it is going to be resistant in other continents, there are certain commands on that databases that you need to prompt, and that one is going to give you the idea of how resistance is going to spread across the country of the world. By now, everybody will know that one of the greatest problems we have in health or in medicine today or in public health is the problem of resistance. And I told you, one of the reasons why this microorganism will continue to develop resistance could be as a result of different reasons. For example, it could be as a result of a drug abuse. When you are not sick, you just take antibiotics anyhow. That is irrational use of antibiotics. When you are using antibiotics irrationally, you are teaching the microorganism, you are teaching them to develop resistance the more. So, and that's the reason why today resistance is still a very great problem in medicine. Then module 5, this is a microbiome analysis using Q2ME2. So and this, you have the step already there. What you do is that you see that guy. By the side of that guy, you can see that the guy is already jotting down something. So what we are saying is, when you are working in bioinformatics, you should be very, 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 very clever to the level that whatever steps you are taking, make sure you document those steps. Because if you are not documenting them, you just discover that at some point in time, you might just get to lose the process along the way. So, and that's the reason why that guy, irrespective of whatever he's doing, he already has this. And when you look at the picture, you will see it's not just ordinary color. All those colors are standing for something unique. They're standing for something unique, and that will go a long way to be able to. So, for example, what that one is doing is microbiome analysis. So if you are taking, for example, let's say you take small soil sample, which is environmental sample. If you take small sample and you want to know the number, the population, the interaction of different microorganisms in that same soil sample, that is what is going to give you the idea of the community. Don't forget that when we talk of microbiome is actually talking about collection of different types of microorganisms, how they exist in community. For example, just like we human, we also have our own community. When we have very similar, or when we have common goals, or they have common goals in the community, there must be interaction. So, and that's the reason why you might want to know the community of microorganisms in that sample. So you don't actually need to do a lot of things. What you just do, if you look at that guy, the guy just has only 2 samples, and from whatever note he is taking, he took note of the location, that is in the soil, the date he collected it, then including the depth at which he has collected his sample, he recorded it. So he took sample one, you can take sample one here, and you can take sample one in any other places. For example, now you want to know in the laboratory, you might want to know where. You want to know the microorganism that is actually responsible for oil spillage. You should know that the population of that microorganism, when there is oil spill, you compare it with where there is no oil spill, and when you now do the comparative analysis, you'll be able to detect the microorganism that is missing there. You'll be able to detect whether they are the ones that are actually responsible for oil spillage or not. And that is what we are saying. You could apply bioinformatics into health, and you can also apply it into environmental sample as well. And that is module 5. Then the next one is phylogenetic analysis using MEGA. By now, every one of you is expected to have know how to draw a simple phylogenetic tree using MEGA. And what do we do? What meaning can we make from phylogenetic tree? Immediately after you must have gotten your result, whatever your result might be. Just what you do is just screenshot it, upload it on ChatGPT, and ask ChatGPT to interpret the phylogenetic tree for you. It's going to do it. In fact, you can even prompt ChatGPT that you don't know anything at all, you are a novice. You don't know anything about microbiology, you are just a beginner. Please interpret this result for me. And that is what to the extent we have gotten when we are talking of advancements now. So for when you just send your sample, you send it outside for analysis, and all those go and cook up some result. It is only somebody who is not serious that will just collect this result and just say, okay, yeah, this is a result, go and analyze. No, you still have to do some tests. So once I send out any result at all, the first thing is that result will come to me first. So when that result comes to me, there are some things I'm going to subject it to some analysis, some validation to actually detect whether it was a cooked result or not. So the same thing that you can also do. So and that is so this one, just like what you have on the board, what you have on the screen, you will see that you import FASTA sequence into your MEGA, then you follow the step that you're supposed to follow, then you build a tree using maximum likelihood plus GTR plus J. You can also use, and for you to also interpret phylogenetic tree, you know, in your first year I gave you one. You can use ITOR for interpretation. So whatever ChatGPT might have given you, you can also validate it using another one. And that's the reason why it is always good for you to know how to use more than one tool for any purpose. If you don't know how to use more than one tool, you will not be able to subject your result to validity test. So that is exactly what we have to do. Then the next one is metagenomic mining. When we talk about mining, by the time you are mining will actually mean that you are digging, you are searching for any result. So metagenomic mining with MG-RAST. MG-RAST is a tool, bioinformatics tool that you can use for data mining. So, and one thing that I want you to know is that it is not that you must know how to use some tools. There are some tools that you will see that even if you do not know how to use them, go back to ChatGPT to teach you how to use that tool. It's going to give you the step-by-step procedure of how you can use it. And just like I used to tell my students, I used to tell them that your supervisor might not be there, but ChatGPT is always there with you. Your supervisor might not be willing to even give you audience. ChatGPT will always want to give you audience, except you don't know how to make use of it. So, and that's the reason why. That's the reason why there are some of my students that this will be the first time that they will be doing project on drug design. Drug design. So I just gave them that they are going to work on drug design. They come to me, I mean, they came to me, Oga, we don't know how to do it, you give us this. I told them that I've already abandoned you with your fate. I've abandoned you in the hand of able and capable hand that is that can give you succor. When your supervisor has forsaken you, ChatGPT will not forsake you. So they are the one that battle with whatever. My own is just go there. And that's the reason why once they are moving at a point in time, I will want to test them. What have you done? What have you done? Can you tell me the reason why you are doing this? Can you tell me? So what I'm saying in essence is just that you should be able to know. So you might not have had some of these tools. This might be the first time you are hearing them or you are coming across them. It doesn't actually mean that you must have known them. In fact, there are times that, let me tell you one secret about bioinformatics. One very funny thing is that you might use a tool this morning and that tool might not be there in the afternoon. There are different. So then in bioinformatics, there are different approaches to to the same goal. So that is why you might also know other tools apart from MG-RAST. That's what I'm telling you. Apart from MG-RAST. For example, if MG-RAST is proving to be too technical for you, you could also tell ChatGPT to suggest an alternative for you that is simpler and it is relatable to a layman, somebody who doesn't know anything. Okay, so the next thing is module 8, and that is visualization. This is at the point where it is our result is becoming more attractive and more interesting. So, and visualization actually means that it is going to generate a kind of images for you, and it's not going to be ordinary image. Those images are going to be very, very what you need to. So for example, if you look at the image you have on the screen, you will discover that that guy is also pointing at pharmaquid. And when you look at the color of pharmaquid, that one is telling you that in that pharmaquid is there. Okay, apart from that, you also have, so you run on your system. After installation, you will do what is called you format the town. It's not going to be in form of faster. It's not going to be in form of faster. It is going to faster. It's not going to be in form of faster. It is going to be in form. You just click on run. When you click on run, it's going to generate an output for you. And by the time it generates the output for you, you go to step 4, and that is open the interactive section. And when you are going to see a case study of a poultry contamination by using a tracking method. Okay, and so summarily, you know that as a bioinformatician, as well as a microbiologist, you should be abreast. You should be abreast with some of these bioinformatics tools like NCBI. You should know how to use NCBI by now. You should be able to know how to blast. That is, you can do sequence matching. You should be able to know Prokka. That is the tool we will use for genome annotation. And you should be able to know CAD RGI. That is for resistance prediction. And you should be able to know that Q2 ME2, that is for your microbiome analysis. And if you are talking of mega, you know that that is when you want to draw phylogenetic tree. You want to establish some evolutionary relationship about your